CAT, Computer Aided Drug Designing. So before we move on to our program, I would like all to pay attention to our corporate video. Thank you everyone for paying your kind attention. Today for the second day of Sage Winter School, we have with us Vinod Devraji. So I welcome you for this session and thank you for sparing your valuable time. And it's my pleasure and honor to introduce you to all my participants who joined us today. Vinod Devraji is a senior scientist in Schrodinger working with rational approaches on computer-aided drug discovery research field for past 10 years. Completed M.Pharma from Madras Medical College, College of Pharmacy, and Pharmacy Bachelor's from Padmavati College of Pharmacy. From 2011, he had taken care of technical support, tech educator, commercial drug discovery projects targeting diverse therapeutic modalities using Schrodinger computational approaches. As an application scientist, he got excellent networking and vast learning opportunities with academic and commercial customers across the globe. He has represented Schrodinger, giving numerous talks and hold experience in hosting collaboratively different workshop programs and co-organized conferences for different institutes and commercial companies on trading scientific topics. On his current research topic of interest are Python programming, drug discovery, biodata cloud analytics. Personally, he is holding 32 research publications in international publications to his credit, covering diverse drug designing approaches as well. Thank you, sir, for joining with us and guiding our students. Over to you, sir, now.
Thank you for such a wonderful introduction. I greatly appreciate it.
Yes, sir.
And so I just want to check, like, what is the audience? Because I've been into pharmacy and I
So
have
we have with us all like from both bachelor's and postgraduate students.
So I'm just like gonna share the slide. It's a combination of a demo and a slide deck. So I'll be switching on. I think I'll try to share my whole screen and then it will be easy. I hope so. Maybe if you can see my slide deck, is it fine? Is it?
Yes, sir. Yes, sir.
Okay.
Completely fine.
Great. So I want these sessions to be as interactive as possible, and I will try to ask questions, different questions. So why do we like this? This always hated me. Like when I joined, and like it didn't switch on when I was doing my UG. When I reached PG, then I had to ask myself why I'm doing this, what I'm gonna gain after this, and why some company has to pay me. I think this question comes out, right? I hope students, are you getting these questions in your mind, or you feel like, okay, I have joined a course and let me finish it off and then later I will think about it. So any comments, like are you thinking about this, like why you have joined this course, and how are you going to be as successful as possible? Have you thought about all this process?
Student?
Anybody? Yes, no, you just have to type.
Yes, sir.
Or I have to consider that you are still that thinking process has not initiated. So usually this happens, like because we are quite monotonous. We will digest whatever is being given. So that's what I have felt. And our self decision capability is the one that kicks out. Like means it was my family situation. Then I had to think about like how to make money and how to make my company to pay me good. So it was in 2011, and I came across this molecular modeling field, and it was quite. I was very passionate that day in day out I used to talk about this, and I started training people, and it became kind of like kind of a mixed interdisciplinary knowledge which I gained, which I'm going to share it with you, and I'm going to share all my experience that which I've gained in different aspects at smaller level and then bigger level and so on. So this will be the summary. Okay. So I know like you don't require, all of you don't require motivation, but some points I will try to highlight with respect to drug discovery. We will speak on different aspects of structure-based drug design and ligand-based drug design. Don't worry. I will try to keep it as practical as possible, and we'll focus on structure-based separately, a demo, and what are the guidelines for target selection and ligand database that you need to focus on, and what is the recent trends that are happening now. Okay, so we'll discuss on all that so that it was useful. Like when you come into industry, you don't talk outdated topics. So just getting started with motivation. The first motivation is actually my company when I joined. Like I was not aware it was a great collaboration. They have a spin-off company called as Nimbus Therapeutics, and they were able to make a big deal of $1.2 billion. So then the company got amazed. Like, how is this possible that a technology can get this much money? And here we are struggling for 6-digit salaries. Like we are very, I know like the situation of how our production companies, because we always rely on, I think manufacturing more, if I'm not wrong, what I have understood. But on R&D scale, like still we are lagging. We are not getting that kind of innovators who are thinking about drug molecules, who are thinking about spinning off their own companies and so on. But so what is the problem which I face? So I don't want to point at anyone. And what I have seen is students will say, sir, like I have funding problems. What do you think? I don't have money, and nobody is supporting me for research. And and feel like I'm a pharmacist, I'm proud of it, and I will work on that only. So that kind of thought process comes. And you are highly like time dependent. You feel like somehow I have to finish my undergrads, and then you have to get into post grad, and then you some of the students are lucky enough to do some grand projects, but that grand project is also prefixed, like objectives and everything is been decided, so you never get to know what happened. You also lack experience, right? I think you don't know like what is going to happen, and you have to reach your milestones. And many of us are quite guide dependent or teacher dependent still, like, so this is what I face. But what thinking process you require is, you should think like, is my target or whatever project I'm taking, is it commercial enough for my company? So this is a thing. It means you have to think like an entrepreneur. I'll just tell in aspect of drug discovery in specific, what is the therapeutic outcome? Is it commercial enough? Whether it has any existing IP, what are the competitor companies, and what are the studies that can be performed with the drug metabolism and pharmacokinetic profiling? Whether the molecule that I'm discussing, is it novel enough to be considered or proposed? Whether can it have options to do hit to lead optimization, hit optimization? What is the potency? What is the selectivity, permeability? All this question has to come in. Now, don't claim yourself that you are a pharmacist, you are not supposed to do all this. If you want to spin off a company of like, if you just do in your UG or PG, okay, get a molecule which is quite potent enough at a cell line level, okay, you can write a proposal for $5 million also to the companies in European countries. So if you think it's a joke, no, but there are many companies that has been spin off, but they do it at PhD level. So always they think like, I will spin off my company and I'll sell that company, I'll then settle it down. But that process is not happening in India. I don't know why. So do you know what is contribution of India in the drug bank or in approved list? I think.
Vinod sir, sorry to disturb you. Actually, your voice is very low. Sound low.
Is it okay now? Hello.
अभी भी
Hello?
कम आ रही है सर आपकी आवाज.
Let me just check microphone.
Maybe
Is it
microphone
okay now?
far away from you.
Okay, let me just try. Is it fine now? Hello.
Yes, then it's okay, sir. Now.
Okay, I'm sorry. Let me just say that I hope you got the concept, like what I was trying to say. Like, what is
Sir,
the concept?
sorry, are you using headphone?
No, I have removed it. Let me just check. Or do you feel like I can try to rejoin? Is it too low?
अभी बीच बीच में अभी जब आप बोल रहे हैं तो अच्छी आवाज आ रही है, पर स्टार्टिंग में बहुत लो आ रही थी आवाज.
Okay, so is it fine now? Can you check now?
Okay, sir.
Okay, so great. So I was just talking about India's contribution in drug discovery. I don't know if this is visible, right? Like, or shall I hide this? Hold on. Hide it. Okay. So India's contribution in drug discovery. So one drug which I'm aware of is saroglitazar, which is specifically for diabetic dyslipidemia. And this drug is from Zydus cadila, originated from India. But this is not as potent or as famous as I'm proud of. But this is not something that we should, this is not used in OTC or famously by our doctors. And other molecules that usually comes out of CDRI. This is a list. I don't know. There are molecules maybe in the recent years, but mostly these are plant-based derivatives, isolated phytoconstituents. And this usually, if you see, are licensed by bigger companies. The reason they license out is just to kill that. They just license it and they finish the project, like, because it is of very less value, right? Like, so they don't want competitors from India to hit this. So that is the reason happened at CDR, right? So due to which we don't have that many molecules launched. But on the other hand, if you see the number of satellites launched, we are, I think, at our peak, right? Like, I think there are a lot of satellites that has been launched. I think so. So what I have to conclude is nothing but drug discovery is harder than rocket science. So you have to think about it and why it is so. I think you already are aware of the whole pipeline that goes in and how much money is involved for the whole process. So imagine like 18,000 crore. It is like too big amount. Like here we think about minor salaries for smaller companies, but think about like how you can handle these problems. And because of technology, it is very, very easy for an interdisciplinary understanding. Okay, so I'm not gonna talk too much. Like, that the target has to be selected, hit has to be identified, then it has to be optimized to hit to lead, then lead optimization, then you have your preclinical trials. Okay, so the reason I think this also maybe would have highlighted simultaneously, you are supposed to do manage all these multiple properties. It's like our brake system. I don't know whether now pharma B Farm and M Farm have brake system or not, but older days it used to be like if you fail in multiple subjects, then you have to redo the year. Something of that sort used to be there. But in similarly, when it comes to drug discovery, if you notice, there are multiple molecules and you have to pass on all the exams. That is on potency, bioavailability, cytochrome inhibition, selectivity, and hug blockage, permeability, solubility, and so on. In each situation, there are different tools, and these tools are very easy to use. Only just we have a fear that only bioinformaticians will be able to handle this. But it's like playing video games. Okay, so if you like if you're a gamer, like you like okay, think like okay, playing with this, then obviously you will be able to do this. So most of the drug discovery fail because of ligand toxicity, or due to documentation issues, or due to poor target selection. I'm going to talk about all that aspect. And this is the traditional approach where they try to synthesize close to 2000 drug molecules. But that is serendipity, okay, trial and error. We used to do that. But in computational, you have millions of design ideas. And again, you will be ending up synthesizing 2000, but the most efficient molecules. But think about at your PG level, you will end up synthesizing only 30 molecules. That's the idea. And obviously, if you do proper in situ works, you will have better exposure. Okay. So the problem of not able to utilize this information is because we usually don't able to balance this experiment theory and simulation. We feel that computation should just do a magic and all our output should be properly correlating with each other. So that doesn't happen. So that is very, very important. But in many cases, if you handle it properly, the computational method and the experimental methods are in correlation with each other. So that is a good thing that we have to think about. Okay, let's dive into what is the aspect of molecular modeling, like how computational biology or chemistry can understand our drug discovery process efficiently. So this is a interdisciplinary field, if you notice. So it's a combination of multiple domains, and that's what we call it as AI. If you have the knowledge of technology and knowledge of history. Hello, yeah, Pankaj. So I hope. Oh, okay, fine. So if anybody have any questions, you can unmute and ask us. Okay, so I'll be happy. Just watch this water molecule. Okay, so you can see it that at the molecular level, these interactions are predicted very well. At 298 Kelvin, you can see there are 4 hydrogen bonds, but at a higher temperature, this hydrogen bond network increases. And due to that jiggling and wiggling, the temperature rises also. Like if we can see, the atoms get dissociated far away, and due to that only it works out. So there are 2 worlds, like Deccan, like you can see Marvels and Deccan, I think, if I'm not wrong. Okay. But in molecular modeling, you have molecular mechanics approaches, and then you have quantum mechanics approaches. Now, in molecular mechanics, it follows Newtonian mechanics, where we have we have bending, stretching, and then you have charges. But in quantum mechanics, both the nuclei and electrons are explicitly treated. One among the famous theories, density function theory. Okay, so in QM application, there are a lot of applicabilities there. Like you can do single point energy calculation, excited state, solvation energies. You can do relax and rigid scan, geometry optimization. You can do UV spectra. All the spectral characterization you can easily do with computational approaches. I don't need big hardwares. It's very easy. I have to compute quantum mechanical optimization, and then I will be able to calculate the spectra, UV, visible, NMR, IR, and Raman spectra. I can generate it. I can also analyze the electronic level properties like polarizability, heat of formation, pK value, thermochemistry, vibrational frequencies, and atomic properties, and all those things by using quantum mechanical application. But when it comes to molecular mechanics, all is driven by force field. Okay, so the knowledge that is gained by experimental outputs and also by quantum mechanical approximation is being feeded into a force field, and that is helping us in understanding the protein structure. So usually these are the major Newtonian and van der Waals and electrostatic forces that handles harmonic bonds, harmonic angles, and then dihedral angles, and then electrostatic and van der Waals forces. Now, like as a humans, like we have certain degree of motions, right? Similarly, molecules have all these forces, and these are well parameterized inside our force field. Okay, so this is the equation for that. It computes your energy. It actually, if you see, the definition is nothing but force field defines the potential energy of the molecular system, right? If somebody asks you what is your potential energy, you will be able to say, I'm very confident, I've studied this much, I know this much, all those stuff. Similarly, a single molecule, its potential is evaluated by using force field and quantum mechanics algorithm. By understanding all this, then you will be able to understand the breathing and quacking of the binding pocket. You can also understand if there are adjacent pockets or there are adjacent cavities that are created. You can also predict the binding site of the pocket also. You can identify water thermodynamics, structural changes, what are the congeneric series, binding cryptic binding pockets, and how the mutation affects. So all these computational approaches can be handled. Now you may say that, okay, how do I know which approach to make? I think if you have heard about it, there are 4 quadrants. According to your literature review, you could notice that, like, let me just slow it down. Okay. So according to that, you can see there are 4 quadrants. So if you do literature review for your research work, there is quadrant 1, where you are aware of the known ligand and you are not aware of the target structure. Then you have to use ligand-based drug discovery. Then you have the known ligands and known ligands. There's a combination of both ligand-based and structure-based. Then unknown and unknown, you have combinator chemistry, homology modeling. And then you have known target and unknown ligands. We do de novo designing, virtual screening, and so on. So by this, we carry out all the approaches. So the major category is structure-based drug design, where the ligand is evaluated on the with reference to the receptor of interest, and the second is ligand-based drug discovery, like pharmacophore modeling and all that. So in structure-based drug design, which is a lock and key, I think many things would have been already been discussed. But for here, you need to note down the major interaction, non-bonding interaction is ion dipole, and you have hydrogen bonding. You can see which is ion dipole is around 40 to 600 kilojoule per mole. Then you have hydrogen bonding, which is 10 to 40 kilojoule per mole. And then dipole is 5 to 25 kilojoule per mole. And then ion indole and dipole and dispersion. All these values as each of them has. So there are 2 categories of inhibitor, right? If you know, covalent inhibitors and non-covalent inhibitors. So very strong covalent inhibitors are there, which is purely for ionic interactions or covalent bonds that is formed, whereas non-bonding interactions are majorly hydrogen bondings, pi and pi cation interactions. Okay, so this is some examples which I wanted to show. This is the hydrogen bonding, okay, where you can see, and this is your pi cation interaction that is shown in Schrodinger, and this is pi interactions, and here you have all the major interactions. So molecular docking is a process, I think you should know. You do a 3-step process, which is docking. What are the possible poses the ligand can adopt? Like this ensemble of ligand poses. Imagine this is the ligand, and these are the various conformations. And it will try to check the ensemble, like how it will fit in. And then we will perform the scoring, and then we can do. But please note that this will not correlate with your experimental value. This is just to give us a good enrichment outcomes. And here is your filtering criteria as which has the best complementarity. So those targets will be considered. So GLIDE is very famous for this systematic search approach. The systematic search algorithm will make sure it identifies the core molecule and it will perform. Hello. I know. Okay. Anybody have any questions? Okay. So the core molecules are available and there is rotatable bonds all around the molecule. So systematically it will pick each rotatable bonds, and due to that algorithm, it has consistency. Like if you see any freeware, if you try to dock 2-3 times, you will see the change in score, and or you may end up in different pose also. So reproducibility is a big problem. That is the reason in commercial environment, they don't use freewares. And the force field parameterization is also not done proper. So the post prediction in glide, there are multiple modes where it is glide SP mode is there, then extra precision mode is there. So extra precision and standard precision, the difference is the conformational search. The conformational search for standard precision is average, whereas glide extra precision has additional conformational search areas. The conformational search areas in extra precision is wide, and also it has good and bad interactions clearly defined. Like what are the good interactions that are available and what are the bad interactions that can affect your affinity. So this is the famous formula. Okay, I know it's quite difficult, but you could see that the major values. It's like a sum of all the components. So it does not have any unit like Coulombic interaction, van der Waals, lipophilic interactions, hydrogen bonds, metal interaction, rotatable bonds, polar phobic interaction, rewards, and so on. Okay, so all these values will help us in telling. So this just a brief video. Watch this.
सर वॉइस नहीं आ रही.
Sir, is there any voice in the video, sir?
Yes, there is some music, so don't worry about it. So I hope you got the message where it was trying to explain you. This is the protein, right?
Yes, sir.
And you saw like it found a complement ligand first, and then it started doing derivatization. Like if you notice, it has just identified the most complement ligand, and then it does derivatization to get the better affinity molecules. That is what your docking is all about. Okay, and let me just give a demo on that part. So I have my Mastro open. Okay. So I hope my screen is visible. Anybody have any questions from docking? Like, are you clear until now? For and like I'll just show you like what target I'm going to deal with and how to work on it. Anybody have any questions?
Basically, when the docking is occur when we have a target, मतलब we have a protein structure, then we have like ligand as well.
Correct.
What we'll do if we don't know about the target or we don't have a protein structure, then how can we develop a this docking?
Okay. No. See, that is the thing. I think if you remember in my one of my slides, I think I showed you it is not a one-way traffic. It is
Yeah.
like this, right? So when you don't have the ligand, then you have to use ligand based, or like if the ligand is not there, then receptor is not there, then you have to use like trial and error approaches. Like you can use already reported other protein or molecules, similar molecules you can test. So this is unknown is very dangerous. Okay, so it's very tricky. Actually, this we call it as sun rising topics or sun rising targets. But otherwise, computational chemistry can handle all the other problems. So is it fine?
Yes, sir.
Okay. Great. Like I'll just tell you like what will happen. Like, so imagine now, like you are working with 6BQ2Q. I will introduce the target also. Don't worry about it. So this is a protein kinase, okay, essential protein kinase for tuberculosis. Okay, so if you inhibit this target 6B2Q, then then imagine, then that means you will have your molecule, just for example. Okay, now how do I reach to 6B2Q? That is the first question you may ask, right? So how I can, if so, I know my target, that is protein kinase A and B, and what I will do, I will go and search that. I'm just doing a reverse engineering. Like I'll select this A and B. You have to just search it. Let me just type it otherwise. Protein kinase A, and I think A, or I think this is B. So you can see this is the protein is actually multiple PDBs IDs are there. So I'll just take this B now. So you will get many hits. Okay. So if you come down, you can see there are many hits. Okay. So you have to select based upon if the method is X-ray diffraction technique. That is the first thing that you have to check. You also have to check whether the organism is okay. Not Homo sapiens. It should be Mycobacterium tuberculosis. I think, if I'm not wrong. Let me just check with 6B2Q. And not only that, you also have to check whether it has any bound ligand or not. This, can you see that? This is just for an example. I'm just showing it to you. So this is protein kinase 2, which is totally a different target. And you have to keep a watch on the bound ligand. So this is 5EA. And you can easily differentiate. As a chemist, you can easily differentiate. And you have to note down what is its biological activity by studying this article. Is it fine? And then you will designate, like for example, 6B2Q, which we will be working on, okay, today. So this is the target. Like, see, you can see that the organism is Mycobacterium tuberculosis. It's not humans, right? Like Homo sapiens, and mutation is no. And the method is X-ray diffraction, right? And you can see the activity is 9 nanomolar for this assay. Now, for those who have done biological activity, they will know 9 nanomolar means it is like a lead molecule. Okay, only toxicological studies has to be checked, because 9 nanomolar is an excellent molecule, right? So that's what you have to check. And if it is in micromolar, that means it is a hit molecule. Like if you see in the presentation, I will try to show it to you. So you are in this stage. Okay. If it is a micromolar, you are in this stage. Then you will do hit to lead. Then you are in this stage. If it is in nanomolar and then it is non-toxic, then that is the concept of lead optimization. So that takes huge money. Like it's like just a gamble. Like for your project, you can just take it and try it out. Okay. So I will now download this 6B2Q. Okay. And here you can see, if you try to download, you will notice there are multiple chains of the same protein. And how do I know that? We can check here. See here. Like, can you see this? This all chain is same. Okay. A, B, C, D is same. The length is same. The bound ligand is same. Then why to keep all the other chain? So that is the reason in computational chemistry, it's like you have to retain whatever is important and others you have to delete. Okay. So there is a tool called as protein preparation. Okay. So in this, if you see, this is the panel, it opens. And once it opens this, you have to click on review structure. So once you do review structure, it's going to show you all the chains. Like for example, this is A chain inhibitor, right? And this is A chain. There is another component which is OBD. So what is this OBD? Let me just check here. So OBD is actually just a other component which is not critical, but you can see, because how do I know this is a molecule? Because CJJ has the reported activity. Okay. And if you want to know more information on this, you have to study this article. Okay. So that is the important aspect that you have to take care. Now in the protein preparation, now I have to understand, see, this is the 4 chains: A chain, B chain, C chain, and then you have D chain. All the chains are there, and I told it's same. So what I'm supposed to do is just delete all the other chains. Okay. Now for all the chain, because I'm just retaining only A chain, and all the ligand components are also there, so which I have to delete now. B chain. Okay, let me just see. I hope it makes sense, like what I'm doing, right? Okay. So please, if anybody have doubts, and you already know the CJJ is the molecule of our interest, so I will only retain that. So this is the inhibitor that I have to work on. I'm just designing some molecules. My boss has given me some molecules to test on this, to check which is the best molecule, why it is the best molecule, and so on. Okay. So usually the proteins have many problems. Now, what are the problems? You have to check this interaction. So you can see it is a raw structure. It has to be prepared, right? It requires hydrogens, and it has a possibility of missing residues also. It has possibilities of missing loops also. So if I have to explain you what is the missing loop is, I'll just have a protein PDB which has that missing loop, then it will make sense to you. Okay. So if you don't mind, you can see here 3 LAU is there. Okay. So this is the protein. Now this has a gap. So I'm gonna go to window, okay, and go to sequence view. Okay. Now this I'm doing because just to check what is the like why you need to prepare it. Okay. So here is the chain break. Can you see here? Okay. So here is the chain break, and I'm gonna just show this, and I'm gonna generate the ribbons. Oh no, not for that. I hope it's visible. Can you see this? See, please get a feel.
Yes, sir.
Don't be monotonous, because it's not going to help us by any means. Okay. So here you can see the gap is there. So this is there, and this you can see this is a big gap. Okay. So, but luckily for my protein, this of interest does not have this gap. I think 245. No, it seems to have a gap. Yeah, it seems to have a gap. Yeah, this has to be fixed. Call this. They just are very curious just to run their score and get the outcome of their binding energy and proceed. Right? You can see here there is one more gap here also. Okay, these are very minor gaps. So at one go, it has options to check that. Like you can see here, the global settings. Let me just see here. Here. Okay, so here all that issues will be taken care. Like fill in missing loops, it is going to take care, and it is going to take care of deleting water 5 Armstrongs to your ligand molecule. It is going to take care of all your other problems like optimizing the hydrogen bond orientations. It is going to clean up your structures for according to your force field. Okay, so all that options are at one click go. Okay, so it's very, very easy, but you have to carefully assess and just don't trust the software, whatever you're gonna do. Let me time. I will import that. I hope I have prepared it properly. Let me see if so. So this is the protein. Okay, let me check if there are any breaks now. Yeah, I think I have to rerun this. Sorry with that. Okay. Okay. So I think you just saw at one go, like how you can identify major problems, and then you can proceed with your preparation. So let this run. I think it will be over soon. Okay. And I will be able to show you like how it has filled it up. But for the time being, I'm just gonna use this prepared structure. So you can see here this bound ligand, which I have to delete now. Let me just delete the other part so that it's easy for us. Okay, so this is the OB and delete from it. Now somebody asked me like, what if you don't know the binding site? I think the ligand is not present. There are two situations. So both of that I will handle now. I will try to split this first of all and show you how the protein looks like. You can see this is the ligand. Okay, this is the raw protein, which does not know where is the ligand. Okay, so usually the protein surface will be like this. Right, in the video, if you remember, this is the situation, right? And the ligand is bound here. Okay. Now, if you are not aware of your binding site in a protein, which is like a, if the ligand is not there, it is called as apoprotein. Okay. So what you need to do is just take your protein and run a program called as sitemap. So here you go. I'm going to run this. And once you run that, it is going to identify all the tentative sites. Though I know the site, sometimes I run this binding site analysis to see what is the composition of the pocket also. So it is going to give me, I think we just started with protein. I just showed you how to select the protein. And then I just showed you how to like prepare the protein. Now I'm just evaluating the protein. Okay, so this is the various sites that it has identified. Site 1, site 2, site 3, and site 4 and site 5. Okay, now how do I know which site is the best site? Now for that you can load it, okay, and load your ligand, which you already have removed. If you remember, see, it is exactly matching with your binding site. If you ask me what is this colors, okay, what are the colors? Yellow color is hydrophobic patch. Blue color is H bond donor, complementary regions. And red color is, let me just show it to you. So if you see here, this is the complementarity of the bound ligand against the target of interest. And if you want to have a scope of growth, then these dots are the indication of the cavities. So that is going to be used by us. Okay. So this is how we identify. And not only that, in the binding site, we have an excellent scoring parameters, which we call as D score. Okay. So this is the project table. Okay. Don't panic. It's quite easy to use. So if you see here, there are lots of properties and I'm activating only sitemap properties. So here you can see there is a primary important scoring called as D score, whose scoring is 1.029. Okay. So this scoring is very, very like if it is more than then definitely it is druggable. The composition of the binding site is also shown to you in the residues column. All the residues, what are, and there are lots of other parameters like what is the site score, what is the size of the pocket, what is the volume, what is the exposure, enclosure, and also that. So you can decide as which pocket is the best pocket based upon site map. But already we are lucky, we already have a ligand to it. And now I have a problem statement that my boss has told me, okay, we know these are the derivatives. Can you tell me which molecule is the best? So if you see, this is the molecule that has been proposed in my lab. It's a collection of, you can see 19 molecules. Okay. So all these 19 molecules, I'm going to show you a demo as how to dock them. And let's decide which molecule is the best. Okay. So for the demo reasons, we will also consider the native bound ligand also. Is it fine? Okay. So the native bound ligand, along with that, other molecules, I have around 20 molecules, which I'm planning to dock. But before docking, I have to tell where I have to dock. Okay, so I think this is the one which is fully prepared. If you remember, I think I just told you, right? Like there is a gaps and that has to be filled in. So you can see here now, so this, I think the gap is too huge. So yeah, so it has tried to fill it up. Can you see that this, okay? So if the gap, like if the gap is too big, then obviously it will not try to fill it up. I think it's close to 10 residues. This is very, very important, and we can do homology modeling and fill the gap later. Like There is a separate tool for that. If it's like sometimes it fails, like, so in that case, you have to fill the gaps using prime, using homology modeling method. Okay, it's not just when structure is not available, when structure is there also. So we will use homology modeling approach, which I'm just gonna skip for this. So this is the build model, and we have to take the template sequence, and from that we have to fill the gaps. But for the demo reason, because the gaps are not in the binding side, so I'm gonna skip that part. See, you can see here, this is the gap, and here is the another gap, which is far away. So both these gaps are far away from the binding side, so I can ignore it and proceed further. I hope it helps. Okay. So here you go. So these are the requirement. Like, I have a plan of docking around 20 molecules and check with the receptor of interest, which is 6B2Q. You have to go to task. Okay, and before that we have to do the grid generation. Okay, so where to dock in the protein, that is very, very important, that we need to understand. And for that, you have to take the protein ligand complex, and you have to go to browse, glide, and receptor grid generation. So this is very, very important that you need to use. And here you can see the molecule option is there. I can pick the molecule. Not only molecule, I can pick the site also. If you see here, this is the entry option by which I can select site map results also. So I will use this option, that 6B2Q. This I will run and I can get the output. Like, okay, so it has already been done. So I'm not running this. I can go to task, browse, and in browse we have options like glide, and we have this ligand docking. So in docking, only three things you need to remember: where to dock, what to dock, and how to dock. So where to dock is the grid that is being generated by using grid generation. If you click browse, here you can see that there is a grid which has been placed, okay, which has been generated already. And this is the ligands, that ligprep out 19 files, okay, that I have to use and for do the docking. But I forgot to tell you like how to prepare the ligands. So that is also very important. Here is an option, okay, in ligprep. Ligprep does the ligand preparation according to the force field OPLS4. You can see here like all the molecules can be selected, and I have already prepared it, and it will also handle the ionization parameters, as whether it is neutralize or it is ionizer, or is it EPIC, which uses Hammett and Taft methodology. In case your target is neutralized, then you have to use neutralize option. And then in case you have to generate possible states, then you have to use this option or specific pH of your interest. Then in case if you're interested with generate automers, that you can generate, but for the time being, I'm not interested. And stereoisomers also, I have to just generate one, because doing chiral separations is very, very difficult. So what I will do, I will just say 6B2Q, and this leak prep will be taken care, and this will prepare. Let me try to give you more processes so that the calculation runs faster. So this is 10 subjobs, and here I'm going to run. So I think we clearly understood now what are the steps we have done, right? So we have. Yeah. Any questions? I think.
Sir, there is a problem with the student. They will unmute themselves.
I request you to mute yourself. Okay, so the ligand preparation is running now, and we are supposed to evaluate all these ligands for its affinity. You can see here, this is 20 ligands, right? Where the top ligand is the native inhibitor, which will use as a control, and we will talk against this target of interest. Okay, So here is the ligands, and this is different modes of docking like standard precision, extra precision, and high throughput mode. So standard precision, as I said, is an average like which you can start with. So I'm gonna just launch this calculation. Let me just see. And here the docking has been done flexible for ligand only. The protein is kept rigid. But we have advanced approaches like induced fit docking. You can see here, this is induced fit docking and molecular dynamics for simulations. I have some files. If time permits, then I'll be able to show it to you. So let me just check. I think it's okay. So you can see here. Okay, yeah, this is running. The job is running. Actually, already launched one more job, so due to which the machine is a little slow. Yeah, it's over now. So the docking is completed, and you can see here the molecules, okay, which has been docked. Once the docking is finished, you have to analyze the results by qualitative needs and quantitative needs. The most important thing is you can compare the native ligand interactions and then decide whether that molecule can be taken into consideration or not. So let us see what is the native interactions. So if you see here, not this one, I'm sorry. Let me just see this. Yeah. So this is the native interactions, which I'm going to pin now. And for comparative reasons, we will compare like how these interactions it's like there, right? Okay. So we will select the molecule. We have to just select the molecule and carefully pick, carefully pick only the interacting residues of the native molecule. So there are two modes of analysis. I'm just doing the qualitative analysis first. Here you can see there is a plus addition mode for addition selections. Okay, so here are the interacting residues. If you notice here, so these are the critical interactions. Now, why a molecule and why experience of a modeler matters is at this point, like where you have to physically inspect. There are automated options available, but this is a very good way of evaluating it. Okay, so I'm just going to show only this residues. So you can see here, this is the native ligand already fixed. Okay. Now, if you are interested to get similar kind of interactions, now what I can do is I can load one by one the docking pose interactions. Like see here, this is the first molecule. It has been docked. Let me just try to show it. Okay, I'm sorry. I think it is the coordinates have changed, so due to which, like, so imagine now this is the place, the coordinates is same. So we will try to compare the interactions by pinning them one by one. Okay, and you have to evaluate these interactions carefully. So imagine this is the ligand molecule. And this is your residues that are interacting one by one. For clarity, I'm just selecting them only and I'm just trying to show it to you. So imagine, so these are the critical interactions that are present and we will try to show it to you now. Okay, now imagine these are the interactions that are present and we will try to now change the poses where you can see like how it changes. Like you can decide whether to retain that molecule or not. Okay. So just don't decide by score. That's a very blunt approach, not good. Okay. So you have to evaluate based upon the interaction, or you have to evaluate based upon the pose orientation also. Like for example, if you try to do it with respect to pose orientation, you have to generate a surface. So here you can, I have generated the surface. It requires high-end graphics, so please bear with me. Yeah, so here it is. Can you see this? The ligand bound, and you have to evaluate one by one all the poses to see if that specific ligand is bound to that specific location of interest or not. So it's a combination and perturbation, as like if you want to consider only interactions or only pose. Okay, and then the same thing can be visualized in 2D also. Like you can just load one by one ligand, and you can see that interactions in 2D. You can see here. Okay, so what kind of interactions it holds. And not only that, at one go, I can summarize all my interactions. So how I can do that is this interaction fingerprint option. In this, I have to generate the fingerprints for all the molecules. Now you can get at single go, it's going to show you all the interaction at one go. So you can color it, you can see like how it is interacting at backbone level, how it is like which ligands are more interacting and less interacting as per hydrophobic residues or overall context. So all that is, you can see all that molecules will be quite promising to consider. So this is qualitative way of analysis. What about the scoring parameters? Now for scoring parameters only, I have kept a native score, right? Like if you remember, so this is the molecule which I have kept it as for reference, right? So for this, we have to watch only the glide docking score, glide primary docking score. Now you can compare, you can consider that anything that is better than the native bound ligand can be the most promising molecules. So that could be an approach, or you can have a cutoff of -9 and above and consider those molecules also. If you are interested with specific values, like, okay, no, I want to compare the energy of the native ligand and energy of the our molecule of interest, so that also you can do. And or specific to van der Waals energy. So any value that is going to help you understand the logics of active and inactive molecules is the parameter that you can consider. So there is no set rule like above this score is good, above that score is bad. So it's purely with your internal reference, you have to decide. For example, now in this, I can say the -9 is a very great score, but I have other molecules, and maybe it is possible if I select this, it may be inactive also. So as per qualitative and quantitative needs, the selection and rejection greatly changes. So that's what is here. And I think I have launched the enrichment calculations for the same study. Let me just see if I can show it to you. So another validation they do in Schrodinger or at commercial level is enrichment calculations. So for that, I have to have a docking, which I have just done it. Let me just see this. So this is the post which I launched before we were starting the discussion. So it has a combination of decoy set of molecules along with active molecules. Okay. So this has been docked. Like you can see here, it's a combination of like close to 811 structures only docked. But I have given provided a decoy set of 1,000 decoys. Okay. So let's see now how it shows. So there is an enrichment calculator options, and the molecules has to be selected, and active molecules need to be picked up like this. So you can see here, let me just pick the active molecules that I thought were the most promising. So here it is. I picked them. Once I run enrichment, I can see overall how it was able to pick up the actives. Like if you remember, I gave 19 active, right, plus native. Okay, so it at within 1%, it was able to pick around 15 molecules. So that's what we see here. Like you can see the excellent plot as it shows, or ROC plot, like more or less you can see that it has excellent picking. If the plot is in the below line, then it is a very poor. So the statistics, bedrock score, the what is the overall area. So the majorly you try to see how enriched your active molecules are picked up from this study. So that is your enrichment outcomes that is usually processed. And not only that, there is binding energy calculation that is different from docking score. So we have a tool in Prime, which is has option of MMGBSA score. So based upon binding energy also, you can select or reject your molecules. Okay, fine. So I think that's all for structure-based drug design, which I thought like showing it to you, but I will be happy to take any questions and I can proceed with the ligand-based drug discovery approach.
Sir, there is a one question on my personal chat box that descriptors which are used by the Schrodinger is are free accessible or there may be on the purchase software only.
No, obviously, see, because it's a proprietary software, only the master interface is accessible, but all the other descriptors are charged. And Sagar Institute has the Schrodinger subscription, so you don't have to worry about it. If yeah, students can easily access it, and for those products which is not available, you can ask me, and I'll be able to help them.
Okay, sir.
Yeah. So I hope there are no questions. No further questions. Anybody, any students have any doubts? Any questions? Great. Okay. Fine. So I will share my screen now and I'll continue my discussion on ligand based. I will elaborate on some aspects and then I'll show you like, okay. So let me just go about it. Structure based is done, right? I hope all of us understood that part. So now ligand based drug design. Okay. So in ligand based drug design, okay, so there are two major techniques which is quite famous is pharmacophore modeling and 3D QSR. So these are the major differences of pharmacophore. Pharmacophore is specifically for new scaffold identification and hit identification. And 3D QSR is specifically used for optimization studies like hit optimization and lead optimization. Pharmacophore is used for exploring various heterocyclic nuclear compounds, and 3D QSR is used to retain same heterocyclic studies done like that. The biological activity is not mandatory. Biological activity is must, and in pharmacophore it is feature-centric screening, and in 3D QSR it is contour-specific screening. A pharmacophoric feature is an ensemble of electronic features according to IUPAC. That is just to ensure like the minimum features required to evoke a biological response, supramicromolar. It could be nanomolar or picomolar response. So as an analogy, if I have to explain what is a functional car, what is the common features for a functional car? So we have wheels and then connecting rims, and then we have the engine, then we have the door. So these are the minimum features to call something as a car, right? So if something has this, then obviously that is a car. In similar manner, how do you compare the structure? So usually it is done by binary fingerprint, chemoinformatic approaches. So when you see this structure, right, structure 1 and structure 2, so what is common? Like if you see, it will look like even for a chemist like me, it seems like different. But they have negative ionizable group in common. They have H-bond donor group in common. They have aromatic groups in common, and then they have hydrophobic group in common. So this is the common features, but the drawback is that if you try to align them, like they are more or less not aligning properly. You can say this is negative ionizable, this is aromatic group, this is H bond donor, and this is hydrophobic group. And when you do conformation search, then you will be able to see that they are aligning properly. So conformation search of molecules is very, very critical. In Schrodinger, at one go, it can generate pharmacophore for multiple ligands. It can generate pharmacophore for single ligand, or it can generate pharmacophore for receptor pocket that does not have bound ligand. It can generate pharmacophore for protein ligand complex. Now, for the demo reasons, I will show only for the single ligand and also for the protein ligand complex, because we already did with 6B2Q, right, which we have prepared. Okay, so just imagine, so this is the protein that you have prepared, and you need to prepare a e-pharmacophore for this. So you have to go to browse, and in browse we have this option of phase, and the phase has options of develop pharmacophore hypothesis. So this is quite smart, like, because it identifies the single ligand and it has shown you that what are the possible features that are possible with this ligand. You can see here, or I think I can just load a ligand and then I can generate a pharmacophore and show it to you. Let me just show it to you. Okay. Imagine, but usually this ligand-based pharmacophore for the receptor is done for the uh, uh, ligand bound, like crystallographic ligand only, not the dock ligand, because this is experimental input. Okay, so I will just change this to protein receptor ligand complex, and you can see this hypothesis can be generated. If you have derivatives of molecules, then you can use these options of multiple ligand, and by that you can generate pharmacophore on that. So let me just try to show it to you. So now the pharmacophore has been launched. Like for single ligand, how does it show? So that, like, imagine now this is the ligand you have. So for single ligand, so these are the features that are available. You can see there are lots of features, right? We don't know. So randomly, like, it's a trial and error when you have just a single molecule, but when it is a bound to a particular crystallographic structure, then you can generate this receptor ligand complex. So right now the calculation is running. If it is like individual, then I can select, like, if this donor is important, then it is knowledge based hypothesis generation. So according to biological activity, if I know these are the important features, I can generate a pharmacophore with that also. So pharmacophore is nothing but these features. So these features have specific distance. You can see this is the distance, and then it has specific angles also, like, and by that the screening occurs. Okay, so if you have a specific features of interest, then the pharmacophore, this distance and angle, perturbation and combination of all the features will help you in screening new molecules. So don't worry, this all options are very, very easy. It's like playing video game only. I just do it day in and day out. So yeah, I think the job is done. I will show you how the receptor based excluded volume looks like. So you can see here. So this is the blue color balls are nothing but receptor based excluded volumes, and I will take that. Hold on. So let me try to load that receptor, then I can show you how this feature matches with the bound ligand. I think the entry is too big. Okay. Yeah, so here it is. So I can load this, not this one. Yeah. Anybody have any questions? Please tell me. Can you see this hypothesis is perfectly matching with the ligand molecule? If I remove this receptor, you can see only the features. So this is actually uh, like a symbol of features that can make your molecule active. Now, once this feature is identified, you have to click on this hypothesis and you can go for next steps and add to database screen. So in phase, you can see there is a phase database and project table. So I have a demo database with me. So I will browse that. Let me just show it to you. So usually these database are pre-prepared and we have options for doing that. Let's go. I'm going to screen. So this is ligand-based screening. Like for the same project, I have used now basic docking. I have performed enrichment calculation. So another famous technique is RMSD post. That is at basic level they do it, but yes, you can also do that options also. Like if I have to compare that, like you can see the screening is going on, then I can show you here is the pose 6B2Q. And this is the docking pose. And this is the crystallographic pose, if I'm not wrong. Let me just see. I think both the orientation changed. I'm sorry for that. Then you can calculate the RMSD of the this is the panel. Like I'll show it to you. So there is a panel called as superposition, and this is going to tell you how to do calculate the RMSD. Just reach out to me. The orientations changed, so I'm not able to do it for the time being. But in the meantime, our screening, hypothetical screening has been done. And this is the hypothesis that we gave as an input. And here we have all the molecules that have new molecules that are not being tested against any target of interest. And this molecule, how well it mapped, it comes in the project table. Okay, so it's very easy here. You can just see the, like I think all the properties are well listed. Like you can see the primary scorings, like fitness score or align score, and there are major scorings like screen score, vector score, and volume score. So you could see here the highest molecules which has the best fitness. Like you can see, this is the highest fitness. It is around 1.966. The highest fitness max we can have it is 3. So this molecules can be most promising molecule to further proceed for our studies. Yes, I think in my topic, I also wanted to update you on like what are the fields that are available or which is currently in trend. Let me just show you that part. And I also wanted to talk on what are the guidelines for target and ligand databases. Okay, so usually we have to select target that is highly commercial in nature, like usually cancer or lifestyle disorders. I think that is well known. And usually you can validate your target by going to clinicaltrials.gov website. Don't just trust on literature alone. So this website has positive and negative case studies. And you also have to check with the template and homologs. What is the sequence view? What is the domain view? What is the gene that you're working with? What is the pathway it works with? It's same like buying a mobile. Like we check with budget, what is the latest model, what is the availability, what are the future drawbacks and resale. So similarly, whenever you're working on a target, please check with all this aspect and then proceed further. And to select a PDB, I think I already explained this, there are so many PDBs and we usually select the organism of our interest, and X-ray is the structure of our preference. Okay, and usually resolution is less than 2.5 Armstrong and it should not be a mutant. That's what I told you. And if it is a bound protein, it usually like we try to predict by using binding site sitemap. And I think these are the problems of the protein. Okay, like you will have chain breaks and you have water molecules. So all this has to be prepared. And there are different possible binding site also. Like this is shallow binding site, which is preferred by peptides. And this is organic binding site. Okay, so which is quite narrow in nature, quite rigid in nature. So this is preferred by organic molecules. And for chemists, the organic molecules are available in different databases and also as a synthetic databases. So binding database is an excellent database for label database for ligand-based drug discovery. You have biological activity also there. Drug bank has all the PKPD testings and all the studies that has been performed against your target of interest. And PubChem, I think everybody knows about this. So, and one of my very interesting database that you can focus on is MolePort. Here, the molecules are directly purchasable. You don't have to synthesize them. You can think about skipping the synthesis. Zinc database is also very well known by many, and medicinal chemists have the knowledge to synthesize novel molecules. So there are tools for automating that also. There are options for enumerating based upon synthetic route options also. So bioassociative replacement options are there. So geometry optimization was performed, and you can see stereoisomerism can be taken care. Ionized and unionized aspects can be taken care. So all these aspects greatly help us in deciding important molecules for our study. I think this is the right time that I have to explain you what is the industrial now recent trends that are happening now. This is very basically like everybody will show you, but there are different other modalities that we have to focus also. One of the modalities is covalent docking. After COVID, I think everybody is thinking about covalent inhibitors because it has to kill the virus, right? So, and the next is macrocycles. This is cyclic peptide. So this is also very important. If you have capability to synthesize this molecule, you will be immediately hired. Okay. And if you can work on DNA encoded library, that means you will immediately be hired. I'm very sure in academics, like very less projects are involved in this. This is de novo fragment-based drug discovery approach. This is also very restricted. Not much people use it. This is again protein degraded, like multiple protein targeting, multiple protein degradation. And this is also a modality that is quite famous. And we have a combination of pipelines by which we perform this virtual screening. So just don't trust on single approach. That is very, very important, where you have to use multiple tools and then get your molecule done. So that's all what I felt like discussing. I hope, I don't know how much time I took, but it was great discussing all these aspects. But it will be interesting to see what kind of questions I get.
Thank you, sir. Participants, the forum is open for question and answer. If you have any query, please let me know. All participants are requested to please turn on your video. So there is an interactive session for half an hour. Sir, there is one question from my side.
Okay. Yes, sir.
Is this possible to go for homology modeling with using this software?
Yes, sir. We have this option. So for example, like in this case, like you saw, like it was not able to fill because of the huge gap, then we have to use homology model to fill those gaps also. Or if you want to build an overall model, then that also you need to do it by using Prime. Homology modeling option is available. Yes.
Okay. And what is the challenges associated with the in silico based approaches with when we go for the natural product drug discovery? Is this possible with using?
Yeah, natural product drug discovery is like usually pharmacophoric based. Like for example, like if you have a natural product, the major hurdle is synthesizing them. It's very difficult than normal organic synthesis. So we have to use various ligand-based approaches for optimization and retaining that integrity of natural compounds. But still, the more or less, but that is the thing. So how you try to synthesize it? Like you can design many thousands of molecules, but if it is not synthesizable, it is of no use. So you have to balance the in silico with the experimental outputs. Otherwise, it's just bluffing around actually. Like if you try to get lots of molecules.
Okay, just as you told that multiple ligands docking can be possible with using this software. So what, how can be result will be produced and how can we differentiate that particular ligands are more active than other?
No, as I said, there are qualitative ways of comparing it with control molecules, right? Like, so control molecule is giving so and so interaction. You can use that as a measure. If control molecule is giving so and so docking score, so that is quantitative measures. You can use both ways for selecting your molecule or rejecting your molecules, but have internal reference. So there is no said novel score or best score, highest score. it's not possible. Yes, in case when you don't have a control, then more negative is better, but otherwise usually try to search for reference molecules.
Uh, for the beginner, I think more of the participant from the B forms. So for the beginner, this drug design software, Schrodinger how can help to improvise the drug by better stand by simple way.
In simple understanding, I think obviously like they would have I think in final year and third year, they have advanced medicinal chemistry.
Yeah, yeah.
If I understand, like obviously means obviously they have to start using this. There is a free academic master available, and you can explore with that interface, and you can check.
Okay, participant, please turn on your video. I think Vinod sir is online.
Yeah, it's okay, no problem. I can understand. Everybody will be in different places, and obviously they will be learning this when they will be in search for the jobs later. Whoever has watched it, because at the end of the day they have to come to market. That happens.
Rajesh sir, I think unmute. Rajesh sir, if you have any question.
Yeah. Anybody have any questions other than this?
You can also type your question in chat box. So can I read that?
No, usually like I'm very lucky. Like many of the workshops, the questions are very less. So I know like our students are quite brilliant and they understand every aspect of it.
Hmm. Actually, B.Com label, the curriculum is not like that कि वो सारी चीजें understand कर पाए इस चीज को.
नहीं, understand. See, as per my understanding is goes, it's like they understand very complex, but it's just like they have to get used to it or have some affinity towards the topic also. So it varies actually. Like so, some student would have attended, they will get back to you asking for more questions. But otherwise, for them, it's just like passing clouds. So we cannot blame them. So they may have other interests or other topics of interest also. That is also possible. So maybe when they are, especially if students get more curious when they are in their final year and curious about their projects. As per my understanding goes like, so that is what. Otherwise, they are quite busy with their curriculum. They focus on the curriculum more.
एक्चुअली सर मेरे समझ में ये आया कि भी यदि मेरे पास प्रोटीन का स्ट्रक्चर है विद आई डाउनलोड फ्रॉम पीडीबी डाटा बैंक एंड देन वी डोंट नो द एग्जैक्ट कैविटी या एग्जैक्ट इंटरेक्शन साइट.
हाँ सर.
पॉसिबल जो भी अभी भी पीडीबी में स्ट्रक्चर्स हैं वो ऑलरेडी स्पेसिफिक बाइंडिंग साइट के साथ में अवेलेबल है कि यहाँ पे आपको बाइंड करना है जिसकी वजह से आपको ये एक्टिविटी मिलेगी. But our drug, our ligand is not like that. They shows the activity. So
No, sir. Like for example, अगर आपका टारगेट एक है और समझ लीजिए ऑलरेडी एक रिसर्चर ने उसी टारगेट पे बाइंड करके नैनोमोलर मॉलिक्यूल दिखा दिया है, तो यूबीएसली ऑब्वियसली यू कैनॉट क्वेश्चन दैट कि और कोई भी साइट हो सकता है क्या, क्योंकि उसने नैनोमोलर में दिखा दिया है. तो लिगैंड टेंटेटिवली, मतलब ये तो वही बात हो गई कि आप एक फोन लेने जा रहे हैं, आपके दोस्त ने बोल दिया इस शॉप में मिलता है चीप फोन, तो आप क्या करेंगे? फिर से जाके सारे शॉप नहीं ढूंढेंगे ना, आप जाएंगे सीधे शॉप में और चीप फोन लेके आ जाएंगे, मतलब कम कॉस्ट पे. तो यही है ड्रग डिस्कवरी में भी, अगर आपका बाउंड लिगैंड अवेलेबल है तो क्वेश्चन नहीं कर सकते फिर से.
और क्या प्रोग्रेस है सर? कंप्यूटर एआई का कितना इन्वॉल्वमेंट है?
एआई तो बच्चों को मतलब ओवरऑल अब फार्मेसी भी सीखना पड़ेगा, कोडिंग भी सीखना पड़ेगा, डेटाबेस भी सीखना पड़ेगा. नहीं तो अभी अगर विदाउट टेक्नोलॉजी जॉब्स अवेलेबल ही नहीं है, मतलब बहुत कम वैकेंसीज निकलती हैं गवर्नमेंट और गवर्नमेंट फार्मेसी कॉलेजेस या सेंट्रल और स्टेट यूनिवर्सिटीज में. तो इट्स अ ह्यूज कंपटीशन. ऑब्वियसली दे हैव टू लैंड इन प्राइवेट जॉब्स. इफ यू आर क्यूरियस अबाउट प्राइवेट जॉब्स, स्टे अपडेटेड. अगर आपको मालूम है वैसे टॉपिक्स तो आराम से ले लेंगे. जैसे मैं बता रहा था, बहुत चैलेंजेस हैं जो जो जैसे प्रोटीन डिग्रेडेशन में और मैक्रोसाइकिल्स में और ये सब टॉपिक मतलब अभी तक मतलब मेन स्ट्रीम में भी नहीं है, पब्लिकेशन में भी नहीं आते हैं. लेकिन इस टॉपिक में अगर जानते हैं तो तुरंत ले लेंगे आपको, तुरंत, मतलब बात ही मत करो आप, वैसे मिल जाएगा। और आप whatever salary you expect, it will be given, and you will have international opportunities. Otherwise नहीं, okay fine, I will be fine with whatever comes in, and also then it's very scope is very less. So it's up to the students as how they take it.
Okay, thank you sir. और कोई
Sir,
क्वेश्चन है?
excuse me, sir. Shall I ask?
प्लीज सर, प्लीज.
Yes sir.
सर, ये मास्ट्रो जो है, ये स्ट्रक्चर विजुअलाइजेशन के लिए इस्तेमाल कर सकते हैं क्या?
हाँ सर,
फ्री,
कर सकते हैं सर. हाँ,
फ्री में?
है सर. हाँ सर, हाँ सर, कर सकते हैं सर आप.
अच्छा अच्छा अच्छा.
तो मैंने एक और क्वेश्चन देखा कि सॉरी, कैन वी परफॉर्म दिस ऑपरेशन इन पाइमॉल सॉफ्टवेयर? नहीं कर सकते मैम. पाइमॉल भी एक जस्ट विजुअलाइजर है, लेकिन मास्टर विजुअलाइजर के साथ कंप्यूटिंग सॉफ्टवेयर भी है. सर आप बताइए सर. हाँ सर. हेलो.
जी सर.
आई थिंक राजेश जी, उनके पास एक और क्वेश्चन है कि बस?
राजेश सर.
जी हाँ, वही पूछना था मुझे मास्टर के बारे में. और दूसरा ये पूछना था.
हाँ सर.
क्या ये जो स्ट्रक्चर बेस्ड या लिगैंड बेस्ड जो डॉकिंग किया, तो ये बहुत मेरा मतलब कम समय में आपने उस स्ट्रोडिंगर के उस मास्टर में दिखाया, लेकिन वो
हाँ
कुछ भी नहीं समझ में आया सच में.
नहीं, मैंने देखिए सर, ये तो जस्ट डेमो है. मैं भी अगर आपको इंटरेस्ट है तो मैं वर्कशॉप्स के लिंक बता दूंगा आपको. पूरे दिन का यही चीज हम पूरा दिन स्लोली बताते हैं.
जी हाँ.
आपको और अंडरस्टैंडिंग हो जाएगी. मैं भी मतलब सागर में भी हम वर्कशॉप कंडक्ट कर सकते हैं. अगर मैंने ऑलरेडी भी प्रपोजल दिया है नीलेश सर को, और आई थिंक वो वर्कआउट हुआ तो आपकी जगह पे फुल डे वर्कशॉप करेंगे. ये तो मतलब जस्ट आपको एक टिप ऑफ द आइसबर्ग. अब वो पूरा मतलब दाल मखनी का बस एक टेस्ट है. अब पूरी दाल मखनी बनानी है तो टाइम तो लगता है ना सर.
जी हाँ. जी हाँ सर, बिल्कुल बिल्कुल.
सर आप हमारे पार्टिसिपेंट को बता पाएंगे कि ये स्टोरेंजर सॉफ्टवेयर यदि वो लिमिटेड डिस्क्रिप्टिव्स के साथ में यदि यूज करना चाहें तो कैसे वो करेंगे, कहाँ से डाउनलोड करेंगे, और ये एक्सेसिबल है या नहीं है?
आराम
मतलब
से सर, मैं वो लिंक अभी शेयर करता हूँ. अगर बच्चों में इंटरेस्ट है या मतलब धीरे धीरे वो यूज करना चाहते हैं,
ताकि वो अपने लैपटॉप पे या अपने डेस्कटॉप पे उसको डाउनलोड करके इट विल बी ट्राई कि उसको कैसे यूज करना है.
आराम से मैं वो लिंक शेयर करूंगा. उनको एक छोटा सा लॉगिन क्रिएट करना है. वो बस वो अपना ईमेल आईडी डाल के अगर लॉगिन क्रिएट करेंगे तो
क्या
दो
आप
दिन
स्क्रीन सर के शेयर करके दिखा सकते हैं सर किस तरीके से उनको प्रोसेस करना है?
ठीक है सर, मैं बता देता हूँ आपको, रुकिए.
Ruben, you can unmute yourself. Can ask.
Thank you, sir. Vinod, sir, is there any resource for our students so that we can just practice this, what you all demonstrated to us,
हाँ सर.
that it would be more clear to us?
ओके, नो ओके. सो मतलब देर आर मल्टीपल वेज. लाइक एक मैं फ्री वाला बताता हूँ और अदर ऑप्शंस में बताता हूँ. तो ये है एक साइन इन अकाउंट है. लाइक अगर आप देखेंगे रजिस्टर नाउ करके आएगा फ्री मास्टर का. क्योंकि मेरा कमर्शियल लॉगिन है तो ये मतलब ये काम नहीं करेगा अभी. मेरे को अगर एरर देगा, मेरे को खुद ही एक्सेसिबल नहीं है. ये यूजुअली एकेडमिक्स के लिए ही फ्री है. देखिए मतलब जैसे आपके लिए बोल रहा है, लेकिन आप लोगों के लिए दिखा देगा कि क्या करना है. इसका मैं अभी आपको चैट में दे रहा हूँ. ओके, ये है फ्री मास्टर आपके लिए. तो इसमें लॉगिन क्रिएट कर लेना, और उसके बाद जैसे लर्निंग के लिए मल्टीपल ऑप्शंस हैं. एक हम वर्कशॉप कंडक्ट करते हैं, पिछले साल भी हमने किया था, और सागर के साथ हम अभी फिर से हम कंडक्ट करेंगे. यहाँ पे एक और है ट्रेनिंग. रुके, ट्रेनिंग. हाँ, तो अगर आप देखेंगे तो यहाँ पे एक एक्सेस मतलब सॉफ्टवेयर $45 का एक है. ये देखिए लर्निंग, बहुत सारे मटेरियल्स हैं इसमें. ट्रेनिंग विद श्रोडिंगर, और बहुत से वेबिनार्स भी चलते रहते हैं, हम भी कंडक्ट करते हैं. तो आपको आउटकम मिल जाएगा उसमें से. मैं आपको एग्जैक्टली वो डिटेल्स बताता हूँ. मैं सर को ईमेल कर दूंगा. और बहुत से ओपनिंग्स भी हैं दैट अभी इंडिया में बहुत से हायरिंग होने वाली है. तो अगर बच्चे थोड़े स्मार्ट हैं और देख लिया तो आराम से आप आ सकते हैं हमारी कंपनी में। बहुत सी ओपनिंग स्पेसिफिक टू इंडिया में है। आई थिंक यहाँ पे साइंटिस्ट के और लोकेशन, लेट मी जस्ट चेक। ये रहा, तो ये दिख रहा है आपको। तो ये इतनी सारी ओपनिंग्स हैं और बहुत और ओपनिंग्स भी आने वाली हैं। तो अगर किसी को इंटरेस्ट है, आपको लगता है कि आप ये चैलेंजेस फेस कर सकते हैं, तो देन यू आर वेरी मच ओपन। तो ये फ्री मैस्ट्रो है, हमने वर्कशॉप, मैंने बताया आप हम कंडक्ट करते हैं, बस आपका इंटरेस्ट होना चाहिए। अब इंटरेस्ट है तो सब कुछ है।
Excuse me, sir.
यस
Sir,
सर।
could we just connect with you in LinkedIn? Do you have
हाँ
a
भाई,
profile there?
क्यों आप डायरेक्टली भी मेरे को फोन कर सकते हो। क्यों लिंक्डइन?
Okay.
आई मीन
Okay, sir. Can you
लिंक्डइन।
share your contact details? In that case.
Yeah, I'm just sharing, and you can get in touch in LinkedIn also. In LinkedIn also, I'm pretty active and share
Thank you, sir.
all the jobs, new jobs, so all those things.
Thank you so much, sir.
Yeah. Anybody else having any other questions?
सर जी,
जी राजेश सर। राजेश सर कुछ और क्वेश्चन पूछना है कि मैं आई कंक्लूड दिस सेशन?
एक आखिरी क्वेश्चन सर।
प्लीज सर।
कॉन्फ़िगरेशन पूछना था पीसी का, मास्टर के लिए क्या होना चाहिए?
मतलब मिनिमम ये, मतलब अगर आप देखेंगे कि इसका मिनिमम 4 GB से ज़्यादा होगा तो बेटर रहता है। 4 GB रैम।
जी हाँ सर। विंडोज 10 या 7 में चलेगा सर?
हाँ, विंडोज 10 मतलब मोस्ट रिसेंट विंडोज 10 के अबव ही, विंडोज 10 ही अभी ट्रेंड में है। तो मोस्टली मेरे को एग्जैक्ट कॉन्फ़िगरेशन देखना है। जब आप डाउनलोड करेंगे देन इट्स बहुत छोटा फाइल ही है, लेस देन 1 GB है, और आपको मिल जाएगा उसमें। या मुझे ईमेल कीजिए जिनको भी इंटरेस्ट है उसके ऊपर, तो मैं एग्जैक्ट इंफॉर्मेशन आपको मैं दे दूंगा। ये मेरी ईमेल आईडी है, अगर किसी को इंटरेस्ट है तो प्लीज रीच आउट। अगर आपको सेम इंटरेस्ट है तो आई थिंक, सॉरी मैं सिर्फ एक बंदे को भेज रहा था।
शेयर कीजिए सर।
9990090044 मेरा फ़ोन नंबर है और ये मेरा ईमेल आईडी है। प्लीज़ अगर किसी को अभी लगता है कि यहाँ, मैं लिंक्डइन में भी बहुत एक्टिव हूँ, मोस्टली आप उसमें भी कनेक्ट कर सकते हैं, और जो भी डाउट्स हैं, क्वेश्चंस हैं, मुझसे पूछ सकते हैं। बट ये मत पूछना कि मतलब ये जॉब नहीं, मुझे स्किल्स के बारे में बात करो आप, क्योंकि जॉब स्किल्स के लिए मिलता है। बिकॉज मेरे को ये क्वेश्चंस भी आते हैं कि सर मुझे इस जॉब के लिए रेफर कर दो, तो आपको 2 मिनट देखना पड़ेगा कि वो स्किल सेट आपके पास है, तो आपको मेरे को पूछने की भी ज़रूरत नहीं है। बट आई कैन गिव डायरेक्शंस की, मतलब ये करो या ये ट्रेंड में है, इसके बारे में पढ़ो। तो इसलिए हम फाइनल ईयर में बोलते रहते हैं। हमें भी बोला था किसी ने और हम अभी पछता रहे हैं तब। सो लर्निंग इज लाइक नेवर एंडिंग प्रोसेस। तो आप अभी से केयरफुली अपने करियर को अगर सही दिशा देंगे देन इट विल बी ग्रेट। और कुछ सर? बस आई थिंक मेरे साइड से तो इतना ही।
Thank you, sir. So I am going to conclude today's session. What a wonderful session by the Mr. Vinod sir. I appreciate that you have spared your valuable time and knowledge with our participant. You are very well explained the target-based and ligand-based drug design by using a Schrodinger software. And I also share the link for the free accessible for our participant. You can learn many activities, many workshop, training session organized by the Schrodinger. I would like to thank our respected chairman, sir, Engineer Sanjeev Agrawal, Vice Chairman Prashant Jain, sir. I also heartful thank to our both ED ma'am, Shivani Agrawal ma'am and Sakshi Agrawal ma'am. for their constant support. I would like to thank our director, sir, Dr. R.B. Goswami, for their encouragement. Thank you, sir. Last, I appreciate every participant who joined and spared their time and joining us for this session. Hope you to see all you in tomorrow. Tomorrow we have our session with Mr. Sant Kumar. He is from ISF Moga. Thank you for all the participants to
Thank you,
joining
sir.
us.
Great. Thank you all participants. Bye-bye. See you then. Thank you.
Thank you, Vinod sir.
Yeah.
Okay, thank you all.
Thank you so much, sir.